Hello, my name is Catherine Simpson. I work for UCB and I would like to thank the organisers for allowing me to speak today. The title of my talk is Incorporating Automation into a Flow Cytometry Workflow for Antibody Discovery, with particular focus on the Biorad Z5. So as I said, I work for UCB, which is a global biopharma company with headquarters based in Belgium. Our main area of focus is generating therapeutics to treat patients suffering from chronic neurological and inflammatory immunological diseases. We have numerous hubs around the world, but we have three strategic research centres located in Bren in Belgium, in Boston in the US, and in Slough in the UK. And sunny Slough is about 30 miles west of London, and it's the technology based at this campus that I'll be talking about today. So I thought I'd start this talk by discussing the antibody discovery process at UCB. Once a target has been identified, and this is usually a protein, animal models are immunized with this protein expressed on either beads or cells. The animals generate an immune response. We then harvest the immune tissues and generate a source of B cells. And we predict that about 1 in every 2,000 B cells will be generating an antibody specific to the antigen of interest. So the B cells are then cultured for approximately 7 days with the view of expanding the B cell population and allowing them to differentiate into antibody secreting cells. And then the antibodies are released into the supernatant. And these kind of experiments are quite large. We're looking at 100 million B cells that we would like to screen, and that can be about 500 plates per experiment. We can then deploy a range of platforms to screen for specific binders, but typically we use bead-based assays. So this is where we have a bead with the target of interest immobilized on the surface of the bead. We hope that we're going to have an antibody that binds to that particular target, and then we can actually detect that binding by adding a secondary fluorescent antibody to allow us to be able to see that. The selected antibodies then go through a round of secondary screening that may include functional cell assays or testing for neutralization, agonism, and binding specificity. And then once these antibodies with the most desirable characteristics are picked, the genes from these B cells producing them are recovered for further engineering and refinement. Some of the platforms associated with this part of the pipeline are shown on this slide, and you may have noticed that there's been a reference at the bottom of these slides. And this is to direct you to a YouTube site that is basically a video and a summary of the process that I've discussed now. And it's a little bit more comprehensive. And I'd encourage you to visit it if you'd like to get some more information. But flow cytometry overlaps with this discovery process that I've just described, and it's a major tool for research into potential diseases that we're trying to target, as well as testing and validating therapeutics that we're developing. And there's three main areas that deploy flow cytometry at UCB. There's that antibody discovery area that I've kind of just discussed. There's in vitro pharmacology that does a lot of the subsequent testing. And there's the therapeutic area that looks at targets before they move into the pipeline and are involved with assessing therapeutics as they're moving towards their more final stage of refinement. So the requirements of these areas are quite different as well. So antibody discovery tends to use smaller volumes and is truly high throughput. We're looking at 384 well plate formats for this area. In vitro pharmacology, again, is still high throughput, can have more variable acquisition volumes, again, medium complexity and low to medium parameter numbers. Whereas the therapeutic area kind of covers all bases. We can look at medium to large volume sample volumes, but we tend to err towards higher parameters and higher spec instruments for deeper phenotyping and assessment. So I'd like to just mention the cytometry platforms that we currently have on site. Antibody discovery and in vitro pharmacology deploy IntelliSite IQ platforms. The robot interface and the software support low sample volume, high throughput screening. And these platforms work quite well. In the therapeutic area, we deploy instrumentation ranging from spectral analyzers and high-end multi-user instruments through to IVD or in vitro diagnostic registered instruments. These have capacity for high throughput in that they can accommodate 96 and 384 well plates, but they don't have a robot interface. So the two groups are kind of mutually exclusive. So we were looking at instrument options for the TA back in 2018, and we not only wanted to expand the instrument portfolio to kind of satisfy the demands of the user base in this area, but we were also mindful of our ability to overlap with other departments and augment the potential of cross-department functionality. So we were looking for a system that was complementary to our current high-end platforms. We wanted a high number of parameters, and this is kind of linked to usually multiple laser lines. We want it to be high throughput. The system had to be able to cope with large data files. And again, we really wanted to deploy a system that had the ability to have cross-department functionality. And the perfect bridge for us was the Biorad Z5. We had a fantastic demo of this system when it was still called the Yeti, being developed by Propel. When the sales rep at the time showed up at our site with the instrument in the back of his car, put it on a trolley, brought it in, set it up on the bench, and it was up and running with, you know, fantastic spec within an hour. So that, to our mind, was a terrific demo and a fantastic endorsement. And you can see here that it started to tick all of the requirements that we wanted from our instrument. But it had lots of other features that were really attractive to us. The automated elements for the shutdown and start up, for that matter. Seamless transition between media options. We can look at plates and tubes and deep well plates. We can control the plate cooling, and it has a really efficient way of mixing samples on plates and tubes, absolute counting, and clog detection. But of course, the real selling point for us was the robot interface. So we invested in the ZE5, and we also involved Propel Robotics in terms of providing a robot solution for us. Now, over the next couple of slides, I'll have graphics on the left-hand side, and then I'm going to have some pictures on the right, and I hope it's going to give you a sense of how the system fits into our lab. So this is a bird's eye view. We have an incubator here. Our incubator is basically a glorified refrigerator. We don't have CO2 attached to it. You could do, but that was our preference. We have the barcode reader here for reading barcodes on the plates, and we also have a row of plate stackers here. The lid removal station is here. This is the robot arm. We have the actual instrument sitting here in the corner, and this is where the operator sits with the monitor to here. So I will have a large arrow appear on the left-hand side here, and that's kind of directing you to the perspective that I've used to actually take the photo of it in the lab here. So this is how it looks. So this again is looking in this direction. So I've opened up the back of the fridge here so that you can see the plate stackers. There's two racks here that hold 44 plates. And on the other side, you can see the interface between the robot arm and the entry port to the incubator. This is a photograph looking back in this direction. You can see again the entry port for the incubator, the barcode reader, and the lid removal station, where the robot places the plate on this location here, removes the lid and places it here, and then delivers the plate to the instrument. This perspective is looking back in this direction here. You can see the barcode reader, again, the lid removal station, the racks, and the entry port to the incubator. And this is basically the same perspective, but looking lower down, where you can see the emergency stop button and the waste containers, as well as the tube racks. An interesting thing also is that you can see that there's no guards around the robot. The robot is very sensitive, and anything that impedes its movement results in a dead stop. So we make people aware that the robot's there, but we didn't have to install any extra safety equipment to accommodate this in a fairly busy lab. And finally, we've got some pictures here of the fluidics supply. So we've got a 25-litre water carboy and a 24-litre waste containment area that allows us to run for 24 hours plus. We can change the waste on the fly, and if we're careful, we can top up the water on the fly as well. So we have had one hardware modification. One of the options offered by this platform is the ability to prevent clogs by performing these cleaning washes using 2% contrad between plates. When running multiple plates, the existing reservoir, which is about 250 mils, is not large enough. So Biorad came up with a solution for us, which was this 4-litre container here. And this was particularly beneficial when running cell screening assays, as the potential for clogs is greater. And so being able to deploy this clog prevention, which I'll cover a little bit more in a minute, was a real asset. So I just want to talk a little bit about the software interfaces that link the robot to the cytometer. There's one that drives the robot is the Ascent software, and it's the Everest software that's the interface for the ZE5 cytometer setup. So if we focus on the Ascent software first, this allows us to load plates onto the plate stackers or indeed into the incubator. We can also tell the software where we would like the plate to return to once it's been in the cytometer. We can ask the system to remove lids, read barcodes. We can order plates and link to an experiment. I've already talked about the clog prevention. We can designate the data destination. We can add personal alerts, and that's when the system emails you when it has a problem. And we can queue and run playlists. So it means that we can have scientists with different experiments queue and then run their samples. So to give you a bit of a flavor of the Ascent software, this is what it looks like when you're first setting up the experiment. You can designate the start and end location for your plate. You can tell the system if you've got lids, and you can ask it to scan barcodes on your plates, which we find extremely useful. Then you can build your playlist. This is one of the best features, and something that my colleagues really like, is the ability to shut down the instrument without having to stay around to do that physically. So we can set that up. We can adjust the grip depending on the type of plate that we're using. We can, as I said, get emails sent to us if there's a problem. I've already talked about the clog prevention, and there's also a clog response that monitors the samples as they're being collected. This is where we allocate the data location, and this is where we add in the plate ID and the experiment template that we want to link to that plate. And so the experiment template is generated in the Everest software, and I'll come back to that in a minute. And then we can set up run lists, and this is where different users can set up run lists, and then we can queue them up to actually run the cytometer when we're ready to go. So looking now at the Everest software, which is the ZE5 interface, and this is where we generate the instrument settings and compensation. We select our media, be that tubes or plates. We can designate the sample volume, event collection criteria, and cooling and heating conditions of the media within the instrument. And so just going through that, we first of all select the parameters that we want to look at. On our instrument, we have 5 lasers and 27 fluorescent parameters and 3 scatter parameters. And while high throughput is plate-based, there's the option for running 45 mil tubes or 40 1.5 mil tubes or 24 1.5 Eppendorf tubes. And changing between media is literally as easy as clicking a button. And so this is where we set up the event collection criteria. Sample collection volumes or event numbers are allocated together with sample acquisition rates. You can allocate when and how long the plate should be agitated and the temperature that it should be kept at, anywhere from 4 to 37 degrees. You can designate the sample acquisition path, which could be down, across, or serpentine, shown in these graphics here. And the probe wash settings can be programmed at this stage to accommodate potentially sticky samples and reduce carryover. So automation supports varying experimental demands, and certainly in our hands and in the environment that I've tried to describe to you already, high throughput does not necessarily in our hands equate to small volumes. And so we can look at the high-end element associated with screening perhaps in antibody discovery, and then for assessing samples and perhaps doing a deeper phenotyping, we might want to use larger volumes and run them at a lower event rate. But it's still high throughput in that there still could be 4 to 10 plates, and it can still take quite a long time to run these samples. So for the Everest software, high throughput mode is very easy. It's literally a slide bar that you push to the left. And then multiple samples are pulled into the sample line with air and gaps in between. This mode enables a fast throughput with very small volume requirements. And you can go as low as 2 microliters per sample, and with a reasonable setup, the ZE5 can finish a 96 well plate in less than 15 minutes and a 384 well plate in less than an hour. And so I'm hoping this graphic kind of explains that here, where you've taken samples, and it's literally just a train, if you like, of samples that are passing through the cytometer. And yes, I've just mentioned that, and yeah, very little carryover. But we kind of wanted to assess this carryover, and we wanted to assess just how good the instrument was at dealing with kind of higher throughput rates. And we thought we would do this using cells. So we fixed some Jerkat cells overnight in methanol. We washed them, and then we stained them with DAPI with the view of looking at the DNA profile. So the odd rows contain cells on this 96 well plate, and the even rows, the rows in between, just contain PBS, with the view that we could then assess the cell profile and then assess the carryover. And then because prior to getting this instrument, I didn't personally have a lot of experience with high throughput systems, I wanted a sanity check to make sure that we weren't getting any misalignment of the wells as we went across the plate. And so in the final row, I spiked in some 8-peak beads so I could have a signature of the alignment. So for this particular plate, I kind of set it up as a standard, in that I used what I would consider a regular acquisition mode. So I collected 40 microliters of sample, and I ran it at 1.5 microliters per second, which is kind of relatively slow, so that I could get a nice CV, so a nice thin peak on my DNA profile here. And I kind of then use this as a benchmark for the other modes that I'll show you just now. So this was the same plate run this time taking 20 microliters at 1.5 microliters per second. And you can see again that we've got nice DNA profiles for our cells. Our wells in between that just have PBS have kind of negligible carryover. And again, the 8-peak beads are seen in the final row there to show that the wells are still aligned. And this was what I would consider a high throughput, low volume assessment. So this was just 8 microliter sips running at 1.5 microliters per second. And again, you can see that the profiles are comparable. The DNA profiles look very good. And again, no spillover in the wells in between. And the 8 peak beads still have 8 peaks, and their profiles are perfectly acceptable for that volume and flow rate. So you might ask, why not routinely use a higher volume at a lower flow rate? And this is where seconds are important. So if you think about an experiment where we have, say, 30 plates with 384 wells, if we add just 1 second per well, we're going to actually add 3.2 hours to the running time of this experiment. So that's why these kind of experiments are important, and why it's important that our system can deal with these kind of settings. And I just wanted to finish off by showing the 8 peak profiles across the lasers. So this was 10 microliter sips at 2 microliters per second, which again is quite fast. And for flow cytometrists out in the audience, you would kind of appreciate that these profiles look good. We've got good sensitivity and good alignment. And that's something that I've been very happy with this system. And just to finish off, the general assessment, I thought I'd put this slide in as well. These are human PBMCs that have been stained with CD4, which is a T cell marker. So we have some cells that are positive and some cells that are negative. And then the cells were collected and the settings on each detector were increased by 50 volts. And we collected it in a range from 350 to 700 volts. And you can see, you don't need to be a cytometrist to kind of see that obviously the detectors are all very linear and are quite sensitive. So one of the interesting outcomes after the installation of this system was how change was inspired to a certain extent in some of the experiment designs. This was particularly evident in larger volume experiments. It's interesting how an extended working day can shape experiment design if you need to sit and feed your plates to the cytometer. Once you introduce automation, including the capacity to clean and shut down an instrument automatically, a whole new world opens up to scientists. And experiments with more replicates, lower acquisition rates, larger volumes become more feasible, and your experiment can run overnight. And we've seen that happen on site. And so I just wanted to kind of give you an example of the other end of the spectrum, where we're kind of looking at larger volumes, slow flow rates, but it's still a high throughput experiment. So this is an example of peripheral blood analysis for stem cells. We're looking at CD45 positive leukocytes here, and then we set up a gating strategy on lymphocytes, and then we copy that strategy and apply it to stem cells. And in this instance, we're using CD34 as a marker of stem cells, and you can see the little guys have popped out here. We apply this established gating regime to this. And I should mention this has been modified from the ISAGE gating protocol developed by Sutherland et al. Quite a few years ago now. And this is the final picture here. So these are the stem cells here that are coloured red that we've kind of backgated. So in this particular sample, they're less than 0.1%. They have a frequency of less than 0.1%. So to see 1,000 events and have something that's relatively statistically robust, we'd need to collect 1 million cells. And that's what we were able to do. So we had 5 parameters, so other parameters beyond the CD34. We fixed the cells. There was 4 plates, 96 4 plates, and there was 57 specimens per plate. We collected 180 microliters of sample at a flow rate of 1 microliter per second, and we agitated every second well. And we had the clog prevention activated. And basically the runtime was 9 hours and 44 minutes, but we were able to get some beautiful data with some lovely tight CVs and numbers robust enough for us to have something that was statistically significant. Right, so I'd like to finish there and hopefully show by this graphic the importance of having excellent support, not only from your colleagues, but also from vendors. And I'd like to thank both Biorad and Propel for their support, and it's been a pleasure working with them. So thank you for listening, and I'll take any questions if there are any. Thank you.